Thank you for coming this afternoon. I'm Sam, and today I'm going to be talking about quality control for single nucleus or single cell RNA sequencing. So,
Sam, please, for the love of God, move that thing.
I'm sorry. Can I minimize? They don't have another screen. They're duplicated.
That's fine.
I'm just gonna move. Okay, there you go. Okay, so here's just like two UMAPs of the same data set. One on the left has like a crappy naive QC that I've done, and on the right is after like a long time of iteratively trying to figure out which are the exact filters that I need to do. And like right off the bat, you can see that these are quite different, where pretty much everything on the left one is like this kind of connected blob. And then on the right, there's more like distinctive clusters, which should kind of correspond to distinct cell types. And this is a mouse brain data set. So we always kind of see the same things in these brain data sets where there's neurons are forming their own clusters, and then the different glia like microglia, oligodendrocytes, astrocytes are forming their own cluster. So this looks like the second I made this, I was like, okay, this is like not great. I need to like keep working on this. So yeah, like how exactly is it that we arrive like at this nice picture on the right? And you know, like what you would see in like one of the Seurat tutorials or the GenPALS tutorials are kind of looking at these like three most basic. I also put this like doublet score here, but you can ignore that for now. The three most basic things to look for are essentially just the percentage of UMI that come from mitochondrial genes. And then another one is going to just be how many UMI do you have in each cell, and UMI by genes essentially. So you would kind of expect like to just be able to remove the outliers might help you, but that's not always the case. Sometimes it's a lot more complicated. I have a question for someone who knows Google Slides better than me. Is there a way to see what slide is coming next? Because it's like,
If you're in presenter mode,
I'm not in presenter mode.
tough luck.
Should I back? Okay. Well, it's gonna be a mystery for me then. It is on there, but it's just like the same image. Right. I feel like it's too late to start doing that, unfortunately. So I'm just gonna like, it'll be fine. Okay. So
Sam, can I ask a question right after that?
yeah.
Can you, so you're saying, aha, this is crappy.
Yeah.
How do you know that's crappy?
Well, there's a lot here that I didn't show you, but like if you see this like big, like, okay, like I was saying here, like you kind of don't expect them to be like all one blob, especially for the brain. Like this, I would consider this as like one blob, right? Like 0, 3, 4, 1, all these ones.
Okay.
Maybe there's a few distinct clusters, but and then especially when I went over here and looked at the percentage of mitochondrial reads is really high in that cluster.
Right.
And then on the outside, it's like definitely just like the number of UMI is like influencing this a lot too. And then, you know, I also did marker genes, and the ones in the middle have like marker genes from basically everything. Like biology, yeah. It's also just like a gut, like I've done, I mean, after you've kind of like done a few of these single cell analyses, you kind of like can tell something is wrong. You might not know what's wrong by just looking at the UMAP, but you can be like, okay, this is abnormal, especially since this is, I mean, the brain is a tissue I've worked with a lot. So you can see like eerie similarities between brain UMAPs for sure.
So visual inspection.
Yeah, and then like double checking like these things and checking the marker genes, they're like not lining up, right? And this is actually after like some naive filtering. So like I think I did a maximum total count of 25,000, and like remove everything with greater than 5% mitochondrial reads. So like this is kind of like what Seurat would suggest you to do, is these basic filtering steps, but clearly in this case, it's actually not enough. Yeah, does that answer your question? Okay. Yo, also with Zoom, would it like pop up if there was an inquiry? Oh, I just have to keep checking.
Yeah,
Okay. Yeah. So cool. Anyways, after, so like the first step, the first like data analysis step is always the pre-processing. So mapping your reads to the reference transcriptome or the reference genome. This can be done as we discussed with like Cell Ranger or Callisto. several different things. A lot of the times they'll give you a quality control report, which is this nice like HTML web page that gives you a bunch of stats about your sample. So I'm gonna just go through a few of those, like for one good sample and one bad sample. So this is the good sample, which is a human brain. So it kind of tells you like, you know, the number of estimated cells, like 3,800. Yeah,
Sorry, can I interrupt you for a second?
Absolutely.
This is brain. Are we looking at single nucleus?
this is single nucleus.
Are the QC steps, you might feel this way, but are the QC steps different if you're looking at single cell versus single nucleus?
They can be different. They're mostly the same. I mean, every data set you're going to want to like tune them like for that data set.
Yeah.
So, but yeah, you can mostly consider them to be very similar. Yeah. So, okay. Like just kind of dissecting this, like it kind of gives you a bunch of different categories. Like, so the big, like the things they put in big numbers are maybe the most important. So how many cells, how many reads per cell, how many genes per cell. These are the numbers that like my PI loves to look at. They'll give you some more like detailed numbers, like how many reads total, how many valid barcodes, and then these like Q30 ones, Q30 is going to be the quality score. So like, okay, 96% of the barcodes had like high quality. Great. So, and then like sequencing saturation, this kind of means like we didn't hit that saturation. So if we wanted to pay more money, we would still be able to get more out of this experiment if we sequence deeper. Whoops. So then you got like your mapping stats. So like 96% map to the genome, pretty good. You know, how much map to the transcriptome and like blah, blah, blah. If any of these numbers were actually really low, which I'll show you later, at least for Cell Ranger, it'll give you this like warning or alert at the top. And oftentimes like these things are so expensive that even if there's a warning or an alert, they're like, okay, you got to just work with what you got, which is like, that was the data set that I was showing earlier. It had a lot of problems. Do you have a question?
Yeah, what do you mean sequencing saturation?
Sequencing saturation is basically like what the point where there's like diminishing returns in terms of like detecting genes or UMIs. So they kind of can like downsample the data to infer saturation, if that makes sense. Yeah. What else? Okay, this plot on the upper right is the barcode rank plot or the knee plot, which is basically on the x-axis is just the barcode. And on the y-axis is how many UMIs. So they're just sorted by UMIs. And this is a logarithmic scale on the x-axis. And you can kind of see like a lot of these knee plots have like, you can kind of see why it's called a knee plot. Like if you think this is like the leg and this is like a knee and this is like your calf or something, right? There's like a clear like inflection point. And that's kind of where we can think of like the inflection, like everything above is where our cells and everything below is going to be background, but it's often not this simple. Like this is quite a nice example where the background barcodes are very easily distinguishable from the real cells. And just to like give you like an idea of what like a good knee plot should look like, on the left is kind of like what I showed you with low background. There's this like sharp drop, and then high background, there's a less sharp drop. So there's a lot of ambient RNA like floating around in each cell. And then here's just a total QC failure where there's essentially like no drop. There's no way to discern like background from noise, but like a lot of the times, like I've been working with like some like reprocessing published data sets recently, like even from like Nature Cell Science, they have like this bad of a profile and they're still publishable. So like you can work with what you've got. You just have to kind of know all the tricks in the books for different QC methods.
Sorry, Sam. I'm
Yeah.
a little lost. Can you kind of break out? So UMI is number of transcripts before PCR amplification, right?
Right.
And then droplet ID is like the barcode that you put on cells, right? Like
This
the
is like
barcode
the
that you
x-axis
use
is basically just like it's like the ranking. So like let's say like the highest number of UMIs out of all your cells is like 20,000 like here.
Okay.
That's gonna have the ranking of 1, and then whatever has like the second most will be 2. So that's the value on the x-axis.
Okay.
Is the barcode rank.
That's fine.
Yeah.
So when you say high background, mean is,
I'll get into it.
it's not totally clear.
I'm getting there. Thanks. Okay, yeah. So I was just showing this like Cell Ranger report, and this is like the first part, but they also have like a mini analysis, and this can kind of give you some idea of how good your sample is also. So this one is going to be a t-SNE, so a 2D representation of the data, similar to UMAP. I think we've already talked about that a little bit. And so on the left is just the t-SNE where each cell is colored by the number of UMI. And then on the right, we have the TC colored by cluster. And like, it doesn't look like a huge, like one big blob, like there is this one big blob, but there's also other blobs. So there's like, okay, cell types are likely being detected here. They also do give you this like marker gene table for the clusters. So like, if you were just like scrolling through, like I can see cluster 1 opalin, this is like an oligodendrocyte marker. And I think cluster 1 was, I mean, it's one of these ones in the middle. So this like is telling me, okay, we have like some, like if you keep diving through this, you'll be like, okay, I see like marker genes I'm expecting. A lot of people don't spend like that much time on this stuff. It's just there if you need it. Also a good thing to send to a PI who might not want to like code it themselves. Now I'm going to get into a like worse quality sample, but it's definitely still like usable, and we're going to like use this in an actual study and not just redo it. So like, instead of just showing you the summary, like the sequencing summary at the top, it'll actually show you these like alerts if there are any. So the first thing it says is like, and like this one is like an angry one. It's like red and it's the error. The other one's like not as angry. It says warning. So the error is like, there's too many reads mapped antisense, like 40% of them, where like the ideal is like less than 10 according to them. And it, yeah, so like this is just like something we have to like think about down the line. I guess like, I think it kind of just like tosses out all the reads mapped antisense, which is weird, but I've seen this kind of show up in a few different data sets. I don't know if anyone else has seen this. And low fraction of reads in cells. I guess this isn't like that far off from what their ideal is, but this is definitely telling me, yeah, it literally says high levels of ambient RNA. And I guess like in a bit, we will go into what that means exactly, but you can think about it like there's a lot of, like when you have your nuclei or cell suspension, there's somehow a lot of just RNA floating outside of cells. So when the droplet forms, like the gem forms with the cell inside of it, there's actually also going to be random other RNAs floating in there that may have come from a bunch of different cells. So we can take that into account with different computational models to try to remove the ambient signal, which is what I'm going to show in a little bit. But you can also just kind of see this like knee plot is like less characteristic of a good one. It's like, this is like high background. So that's kind of still, this is just like informing us like maybe certain steps we should take with this data set versus others that may be higher quality. Yeah, anyways, we can just like keep looking at this stuff and blah, 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 blah. And yeah, here's actually a sequencing saturation plot, which is going to show the mean reads per cell versus sequencing saturation. And like, you can see that this goes up as there are higher reads per cell. So basically, if we were just to pay for more sequencing, then we would eventually hit this point where there's like no more we can get by spending more money. Yeah. Similarly, like SplitSeq gives you a similar quality report, and yeah, it's like extremely similar. They have the knee plot that they give you, and they have the sequencing saturation. They have all these stats. SplitSeq is kind of nice because you can actually just query any gene and then like look at the expression of it. It'll also show you where all the different samples are, since SplitSeq you usually do more than one sample together. And it'll show you clusters. Yeah, I guess I didn't have a good transition for this, but the knee plot, like figuring out what's a cell versus like an empty droplet or a barcode is like kind of challenging. And like SplitPipe has like this naive way of doing it where there's like a hard cutoff. Like they kind of, they have some way of inferring like where the inflection point is. And then it's like, okay, everything on the left is a cell, everything on the right is empty droplet. A few years ago in genome biology was this publication called Empty Drops. And they have like a slightly more sophisticated way of doing it where they like fit some regression. And then they're like, okay, cool. There's like some gray area basically in between here where some things are cells and some things are not cells. I have never used this algorithm, but that's just like one option if you are interested in looking at that. So like kind of like the empty drops knee plot might look like this, where you have all of the barcodes in the top left, and then what they're referring to as empty droplets in the top right. And then so like large cells, so cells with a lot of UMI, and then like small cells. So these things, the small cells with fewer UMI, there's going to be some empty droplets that are overlapping. So some of the empty droplets have like so much ambient RNA that they actually kind of look like a small cell. And this could be just like a small cell can be like a physically small cell. So it just like contains less RNA because it's like literally small, or it could just be like by chance it was not as many reads or sequence from that cell. Something that I use pretty routinely nowadays, especially for those more low quality samples, is called cellbender remove background, which is a deep generative model, which is a fancy word for neural network, for unsupervised removal of background noise from single cell RNA-seq datasets. So they have this like little cartoon of the microfluidics platform where the cells are going into these microfluidic droplets, and the little like lines over here are going to be the ambient RNA floating around. So if there's a lot of ambient RNA in your solution, you're going to definitely see that in the downstream steps, as I've shown you already. You might see like basically cellbender is good for this like kind of knee plot situation where the drop off is like not quite so clear. It can do a pretty good job at determining cell versus empty droplet. And it also actually will like augment the counts matrix to like actually remove counts from ambient RNA in the cells that you keep, which is pretty cool. So the one parameter you actually do have to supply is number of expected nuclei. It's like not super sensitive to this parameter. So like for this one, I just made a knee plot and kind of like manually looked with my eyes like, okay, 2,500 kind of looks like that's where the inflection might be. And then it gives you like the output of this, like one of the visual outputs is just this knee plot, but it also has cell probability plotted on top of it. So like high, like 1 is gonna be like, okay, this is a cell. And then somewhere in the middle is like less confident. And then at the bottom, it's like, yeah, it's definitely not a cell. So you can kind of see like somewhere around the inflection point, this actually might be like a few hundred more cells than I told it, like probably like 3,000 or something, is where there's this like gray area where some things are cells, some things it's not quite sure, some things are definitely not cells. It also will like spit out this kind of progress of like, okay, it is like, yes.
Yeah.
It can take, yeah, so unfortunately I didn't want to include this today in our like notebook because it requires a GPU since it's a neural network. You can use GPUs on Google Colab, but I was not sure like it takes me like for one sample like 20 minutes usually. So you have to give it you kind of tell it like two things, like how many expected cells and then how many like empty or like how many more barcodes beyond that. So like for this one, I gave it 15,000, but some of our like, at least some of my lab's more recent experiments were trying to get up to 10,000 cells. So in that case, I'll give it like 30,000 like total barcodes. So there's like, because it still needs, it's essentially like trying to draw a comparison between things that are confidently empty barcodes, so with very few UMI, and then with like confidently cells, so like really high UMI. Does that make sense? So you need to give it like a good amount of both. There are some samples I've downloaded from like published studies where there's like maybe only like 150 cells total, and then it's like not great at doing those. It just figures like everything's a cell or everything's not a cell, which is not ideal. Yeah. You give it a
Repeat
counts
the question.
matrix. Wait, what?
Repeat the question.
Yeah, Axel was just asking if cellbender works from all type of alignment output or just from like a specific one. And so I've used this for, I've done this with like Callisto output and Cell Ranger output. You just need a counts matrix. It doesn't even need to know like what the genes are. You could just name the genes like anything, like, yeah. Yeah, so it'll tell you like, sometimes like if it's really not doing a good job. So like this is like, the y-axis of this plot is like evidence lower bound, which is kind of just something that we are trying to maximize throughout the training of the neural network. So you kind of want this pattern where it's going up, right? And then it's showing you the training and the testing procedure on top of each other. And then epoch is basically like how long we're training it. So like I gave it 150 epochs, probably overkill in this case because it looks like it kind of levels off after like 50. So there's a few different parameters you can play with for cellbender. It'll also like, since it's an unsupervised generative model, it does give you that like latent encoding. So they'd go ahead and like show you PCA of this latent encoding. And like roughly this should kind of tell you if there's different cell types, but I honestly don't find this one like extremely useful. This is just one of the default outputs that they give you. This is
It's kind of strange that they do a PCA out of a
right, yeah.
blank space.
Yeah, because it's like already, yeah, whatever.
Sure.
Yeah, so this is from this, oh yeah, also this is weird, but Cellbender is kind of one of those papers, it's like in bioRxiv hell, where it's like from 2019, but it's not published. And it's also maybe worth noting that like some of the same authors are from the empty drops paper, so they kind of are like thinking about this topic a lot. And this is kind of like the second version of, I mean, it's like not like computationally that similar to empty drops, but this is kind of like their second round of trying to solve the same problem. But yeah, they're showing these different knee plots, the Cell Ranger V2, knee plot V3, empty drops, and Cellbender. So like right off the bat, you can kind of see there's not much of overlap between this like cell in gray versus like not a cell in red for both Cell Ranger V2 and V3. Also, I think by now they're like on Cell Ranger like V6 or something. So they might have a more sophisticated way of doing this now. But also like for empty drops, like it's probably tossing out like way too many cells, like all the way up here and like really high in the barcode rank is telling things are not a cell. Cellbender is like a little bit more sensitive to these things in the middle. So it's like there's a little bit more overlap. And then they go on to explain, I don't know, the paper's pretty long and there's a lot of math. So if you're not afraid of math, I would go and read it. But yeah, that's all I wanted to, and I kind of wanted to show like myself using it on one data set. So this is just a UMAP of one sample from like the big one I showed earlier. And I also am showing these marker genes. So these are just like hand-picked like known marker genes of these cell types. And like the pattern I want to like you to pay attention to is that like for this gene, like MOBP, it's like there's this low level of expression in literally every cluster. And there's also for these neuron marker genes, there's this low level of expression in every cluster. And this is kind of what I'm referring to with like ambient RNA is like, those are just like UMIs that are everywhere. And then after Cellbender, so we have a different UMAP because we have like basically a different counts matrix we're starting from. But if you look at those same genes, it's like the amount of ambient signal is like very diminished. Like it's maybe there a little bit, but not quite so much. So this definitely helped me clean up that particular data set I was showing at the very start. You have a question?
Yeah, so for Cellbender, do you typically run it per sample or would you run it on a whole experiment then?
I run it per sample.
Is there a reason that you do that instead of running it on an entire experiment?
Yeah, because like for 10x, when the like you do them separately for each sample.
What do you think, what would your recommendation be for the combinatorial barcoding?
I run them per sublibrary.
Okay.
I didn't show any of that though, but it does work pretty well for split-seq or combinatorial barcoding. Yeah, anyways. The next thing I'm going to talk about is doublet detection. Also, wait, how am I doing on time? Wow. Okay. Doublet detection is essentially like this thing that happens when more than one cell gets the same barcode. And this happens quite a lot. For 10x genomics, I think they try to tell you it's about 7% or something. That's like what they're expecting, but that it changes depending on tissue. It changes depending on single cell versus single nucleus. But since they're trying to sell you a product, it's actually really hard to figure out like what the real expected doublet rate is. So fortunately, a lot of people have been working on this problem of computationally figuring out what is a doublet versus what is a real cell. So 2 of these papers were actually published at the same issue of Cell Systems. So Scrublet and Doublet Finder were both published in the same exact issue. And I feel like there are some like algorithmic differences, but they kind of all do this similar approach where they are making like fake doublets. So they take like your cells in your data set and they like artificially combine them. And then they reprocess your data with the fake doublets to figure out, okay, who's neighboring with these fake doublets very often. And they assign some kind of like doublet score based on how many artificial doublets are neighboring the real cells. They slightly riff on it a little bit, but all 3 of these methods are kind of doing that, like simulate artificial doublets and then analyze the data with those doublets. So today I'm mostly going to show you stuff from the Scrublet paper because this is in Python, and so it works well with our other notebook that we've already been working on. Doublet Finder and Doublet Decon are they're both R packages. So this is just like I was saying, this is a little overview of the Scrublet algorithm. It's first in like panel A is just showing you like kind of what's happening in when you're doing the single cell experiment. You have some cells, I think the little gray, this thing is like the little cell. So you have the bubble with only 1 cell, that's what you want. This is the bubble with 2 or maybe even 3 or more cells. You can actually imagine like if it's 2 cells of the same like biological cell type, it's harder to figure out if that's a doublet versus 2 cells of different types. Because there's certain marker genes, like for example, oligodendrocyte and neuron tend to not share a lot of genes. So those are kind of easy to find. And for a doublet or triplet or multiplet, whatever it is, comprised of 2 cells of the same type, the only way you might find them is actually if they have like a really high number of UMI. Anyways, they're just showing this little cartoon where they have these detectable doublets. Those are the ones from 2 different cell types. And then they have these like undetectable ones that are kind of like hiding within the other clusters. And they're kind of showing you, like, I'll get into it, but they kind of show you that this, their algorithm should work whether you're studying a developmental trajectory. So there's like more of a continuum of gene expression versus like a fully differentiated system where there's these like distinct clusters, kind of like the brain stuff I was showing you earlier. And yeah, just like I was saying, the first thing they do is essentially simulate these doublets, and they build this k nearest neighbors graph and calculate the fraction of simulated doublet neighbors to give a doublet score to each cell. Okay, so the way they evaluated this, it kind of makes sense where they have this like, they basically like have genotyped individuals and then they analyze them together so that they know which individual each cell came from. So if they have reads from 2 different individuals, they know for sure it's doublets. So they have some ground truth there. And then they can kind of see how good their algorithm is by seeing how much it picks up on the ground truth doublets. I didn't show it, but I think that they tell you it's like, we did a good job. And then they kind of show something similar on a differentiating cell trajectory. So they're kind of showing you that it picks up on these things like that are kind of lying outside of the trajectory. They're just like all in over here, basically. Okay, so like they're doing their job to convince you. And then I ran this on 1 data set, and you can kind of see like just this little UMAP, and it's like maybe like this little cluster over here, like in blue, is like, those are the neotypic doublets. And then there's a few more just like hiding here on like, they kind of tend to go on like the corners of different of these different clusters. I don't know. It's just like weird patterns I've noticed like when doing these things.
Well, you can see, I mean, going way back to the first box you showed us, like the total counts tend to do that as well.
Yeah, yeah,
So it's probably for the same reason. I don't know what that reason is. It's probably for the same reason. Because if you're doubling, you're gonna have more counts. Right.
yeah,
Right.
yeah. I was actually, that's a great segue because I was just like looking at these different like the kind of like very standard QC metrics, splitting them by doublets versus singlets. The number of genes by counts are like a lot higher in doublets, but and so are like the total counts, so the total UMI. But actually, like, you know, mitochondrial is not going to really change. I guess that kind of makes sense. There's no reason it would necessarily be higher.
Well, that's a percent.
Yeah. Oh,
Right?
good
So
point.
it makes sense that the percent isn't changed. But if you look
The
at
total would probably be
actual-
changed.
Actual numbers.
Yeah. Yeah. Anyways. Yeah. Yeah, I'm gonna get into that. Well, that's actually in the notebook. But like Scrublet actually tries to automatically assign a cutoff. What we will learn in the notebook is that sometimes what Scrublet tells you is terrible. Like they give a score like 0 through 1 or whatever, and then they also try to automatically find the cutoff for you. And so like they'll give you two things basically are added to your AND data is going to be a score and like a Boolean of just true or false. So like if you're like a lot of people probably just trust it and they're like, oh cool, I have this true and false. I'm just gonna like get rid of everything that's a doublet, right? Well, the example I'm gonna show you, it only said there's like one doublet, which is probably not true based on their cutoff. I'm sorry, what did you say? I was trying to look at the chat. Yeah.
Okay, I think you just basically look at the likely distribution for and then for one this one.
Yeah, exactly.
So how can you I don't think that's enough.
I feel like there's,
But just feel it like.
Okay. I didn't put this in the slides, but sometimes like I will just take out entire clusters that have a high doublet score. And that's it. So like, and then sometimes I'll just do something really arbitrary, like 5%. And like, I think that like, a lot of like when you're doing the when you're in the like peer review stage of the study, a lot of the reviewers will like try to think they're really clever and be like, did you do doublet detection? And like they just to kind of like appease them, I would like at least remove like 1% of your data. Like, but well, so what I actually did in my paper, which the whole peer review document is available if you like really want to look at it, but they asked us, we didn't do any doublet detection. And we asked them, or they asked us about it like after like the second round of revision, and we're like, oh my God, I don't want to like redo everything. So what we did is we ran 3 different doublet detection algorithms and we showed that there was very little overlap in the results. There was like almost no overlap, which is like kind of sad, but I think like for the purpose of at least like appeasing the reviewers, it's still worth doing, whether or not that's a good thing. You know, gotta play the game. Yeah.
What I will say though is I've done doublet detection before, and I find that actually some of the data, not all the data, but like some of the doublets actually are very interesting from a biological perspective.
Okay.
Right. So we've seen that, for instance, I look at like an unvaccinated versus vaccinated data set. In the vaccinated data set, we see lots of B and T cell doublets.
That's interesting.
Which is really.
Do you think there's a
biological.
physical interaction?
Yes.
Okay, that's cool. Because like for me, at least, since we're doing single nucleus, like we have to lyse the cells, right? So like there's not going to be nuclei of 2 cells that are like physically connecting.
Yeah.
But I think that, yeah, there may be some interesting biology behind like the cells that are physically connected.
Yeah.
But yeah, this is why like every day there's no one size fits all. Is like the repeating theme of this seminar is like you have to consider what tools are in your toolbox and when to use each one.
Well, I also think, but going back to Narcissus' point, like, you know, you can leave the doublets in.
Yeah,
Like, if you're not screwing with your data.
a lot of papers they'll just have like, there's the doublets right there, like,
Yeah.
but they won't remove them.
I mean, as for multiple data set that I work, for example, then as Sam said, then I used to like the old predicted double this school, I just didn't find any double.
Yeah,
And then for some of the other like, I think I just only find a few, so.
You're probably fine. I mean, chances are, this may or may not be right, but chances are, if they're not obvious, then they're probably not screwing with their data to an obvious extent. So it'll change your analysis, but it may not like totally break things,
yeah,
you know?
yeah. So at least for me, it's like, you would think like, okay, you can use a doublet detection method to get rid of the doublets from two different cell types. I mean, you could probably just use it like a UMI cutoff to get rid of the other ones. But at least for brain people, like neurons have so much more expression than every other cell type that we can't just do the high cutoff because that'll only remove it from the neurons. Like if anything, we have to actually do like some percentile from each cluster separately that we remove. Or each cell type. Like you do some kind of cell type annotation and then like, okay, I'm gonna remove the top, like whatever quantile percentile of the UMI for each cell type. So lots to consider.
You have an actual question.
Oh, wow.
Kind of just answered a little bit.
So Andrew is asking, instead of trying to detect and remove doublets, is it normal to just remove cells with very high gene counts and high mitochondrial percentage? I think that was kind of like, that was, well, like all these doublet papers were published 2019. So before that, this is kind of all there was these cutoffs. And nowadays, I think it's pretty standard to have a cutoff and maybe some other criteria of removing things like, or you can kind of think like, it doesn't always have to be like a fancy algorithm to detect doublets. You might just literally see like, there's this one cluster that has marker genes from every cell type. I'm just gonna get rid of it. But yeah, it's definitely normal to remove cells with extremely high gene counts and mitochondrial percent. The mitochondrial percent thing is actually, I don't even know if I had a slide on it, but a lot of times it kind of indicates the cell is dying. But I know for sure in some systems, especially cancer, they can have a lot of mitochondrial reasons for other reasons. So once again, you don't always want to just remove the ones with high mitochondrial. You have to consider what you're doing. I think I'm almost done. So the last thing I was going to talk about very briefly, and this is not in the notebook because it's not actually, whatever, the ScanPy way of doing this was not as nice as I hoped it would be, is dealing with the cell cycle, because sometimes the cell cycle causes a lot of variability in the data set. So this is just from the Seurat. This is just a picture I pulled straight from the Seurat tutorial on how to do cell cycle regression. So each dot here is a cell, and they're actually showing it as a principal component plot, not like a TC or UMAP. And then Seurat and ScanPy both have these like cell cycle scoring algorithms where it looks at different genes that are known to be involved with the cell cycle, and it tries to figure out which part of the cell cycle each gene is in. So here you can actually see in the principal component space this clear delineation of cells based on the cell cycle. So Seurat and ScanPy both provide a handy dandy function to basically, when you're doing the data scaling, it's going to regress out the effect of the cell cycle. So what that looks like in the principal component space is this. So it's just like a mixed up blob where, I mean, hopefully the different colors are all mixed. It looks pretty mixed to me. This is like, this isn't like a QC, like a removing things, but this is just like oh, I've identified a potentially unwanted source of variation in my data, and this is something I can do to correct it. Definitely not something you always need to do, but it's something you should probably always look for. Does anyone know if there's like a biological reason? Like, are there any systems where like, I guess if it's like developmental system, right, you would definitely not want to regress it out, right?
I mean, right, because so you have development systems, but those cells could be dividing, progressing, not dividing. Do you know what I'm saying? I think.
Yeah. So will it screw you up if you don't regress it out?
Yeah. What?
Just mouse like
Yeah, because sometimes it's really strong, right? Yeah, so at least like in the brain, I almost never do this because neurons like don't really differentiate very much. So especially, like if it was a developing brain, it'd be a different story. But adult brain, probably not needing to do this. Although like one of my projects recently was this like, it is a brain project, but it's specifically looking at oligodendrocytes, and there's always this kind of like stem-like oligodendrocyte progenitor population just like chilling around the brain. And the experiment was basically like injecting the spinal cord to something that's going to kill the myelin. So it's like the myelin's basically this compound that like insulates the axons to help like conductivity along the axons in the brain. So when you remove that, the oligodendrocytes are like, oh crap, I have to like start differentiating and like make more myelin. So like for that one study, I did have to look at the cell cycle specifically for that reason. But yeah, I guess this is the last thing, and I kind of started with this also, is just like the very basic filtering based on just UMI or genes. So this is just like some UMAP I have, and then you can look at it by, how many UMI, how many genes you have, and the percentage of mitochondrial. So like this little cluster over here, cluster 12, is like maybe some outlier because like it's having a giant percentage of the reads that are just from the mitochondria. And you can also kind of look at these things based on the different samples. So this is the data set we've been working with for the tutorials. And like just based on this, you can see like, especially with mitochondria, like, okay, this like, I don't know, some of these samples like C9 has like much fewer than like P2, right? The distribution is very different. You can also look at this, yeah, you can also look at this based on each of the clusters, because some clusters are going to be heavily influenced like by this. So yeah, that cluster 12 has a lot of percentage mitochondrial reads. The ones with more UMIs actually might just be the neurons in this case. They probably are. So like I was saying, it's like really hard to just kind of pick a UMI cutoff, especially for the brain and probably other cell types. But that's enough time for slides.